Hey everyone, welcome to another Mental Dental Live. I'm Dr. Ryan, and we're so glad to have you here. We're going to just open with a few minutes, just welcoming people into this live event. I hope you're having a great day, evening, wherever you are. Thank you so much for joining us. We're excited to introduce this topic, oral microbiome transplantation, and we have a couple of special guests with us who will be joining us shortly to talk more about their exciting research. If you haven't yet seen the newest podcast episode that I released, that came out just last week. Don't worry if you haven't seen it, you can watch it later. But it is there for you so you can watch it at your leisure. And we're going to be talking to the guests that I interviewed there tonight. So if you have any questions about what this is all about, this will be the time to do so. But as you're joining the live event here, I'd love to have you just message in the chat. You can leave maybe where you're watching from. It's always fun to see where everyone is watching from. So go ahead and type in the chat. We'll make sure that you're here and you can hear me. Hey, Gabriella, it's good to see you. Paul, good to see you. I was talking to you in the Discord server not too long ago. Good to see you here too. We have some California people. That's great. Dallas, Texas. We always have Texas representing. I know that for sure. Portland, from Egypt. Fantastic. All over the world. Glad to have you all here. Thank you so much for being here. We have Brazil. Laura, good to see you again. I remember you from the last live event. Great to have you back. San Antonio, Texas, Iraq. Wow, everywhere. This is fantastic. Great. Well, what we're going to do is I'm going to introduce some of the special guests we have behind the scenes here, and we're going to bring them on stage so you can talk and interact with them. This is going to be a very casual event, a very just basically an opportunity for you to ask some questions and get some answers from some experts in this field. So first, I'm going to introduce Kevin Ketagoda. Here's Kevin. And Kevin, you want to introduce yourself briefly to our chat?
Hi, hi everybody. Thanks for joining the Mental Dental live Q&A. So my name is Kevin Ketagoda, and I'm doing a joint PhD with University of Adelaide all the way from Australia and Penn State University here in the US. And I'm the technical lead on this project. And I've got a background in microbiology, immunology, and a bit of biochemistry.
Awesome. Great to have you here, Kevin. We also have Dr. Laura Weyrich. Thank you so much for joining us as well, Laura.
Hi, everybody. Thanks so much for being here tonight. We really appreciate it. We look forward to chatting with you and having time to answer any questions that you might have. So I'm an associate professor of anthropology and bioethics at Penn State, but I'm a microbiologist by training. And I do a lot on the history of our oral microbiomes and really how we might take all that information and translate it into new technologies that might improve health across many different populations today. So I'm really looking forward to chatting with everybody. And thanks so much, Dr. Ryan, for having us on. We really appreciate it.
Yeah, absolutely. Absolutely. And so I guess we could just kind of start in case people haven't had a chance to listen to that podcast with just a brief kind of introduction to what y'all are working on. So Laura, if you want to just talk a little bit about some background to this project and kind of where we're at today.
Yeah, sounds great. So when I started my postdoc, I was working on researching ancient oral microbiomes being these collections of healthy, commensal, often beneficial microbes that live in the body with us and really actually are more than 50% of our total cells collectively across our gut and mouth and everything else. But during my postdoc, I was really researching these microbes in the mouth and how they've evolved over time and space. And so one of the things I was lucky enough to do was actually reconstruct the oral microbiome of a Neanderthal. And one of the amazing things we found is that Neanderthals actually have really great oral health and they have awesome microbes in their mouths. And we really wanted to understand why that was the case and also then track how the human microbiome has changed through time. We know today in living societies all over the world that dental caries or cavities or dental decay, whatever you want to call it, it's fine with me, and gum disease or periodontal disease, these are really can be initiated by microbes in the mouth. And in many countries today, we know, that there are different types of microbiomes associated with caries development. There's different types of microbiomes associated with periodontal disease. But in the past, this wasn't as widespread as it is today, right? Not until we started farming. So we really spent a lot of time trying to reconstruct this history and understand how we got these disease-associated microbes that we have today. And so we kind of used ancient DNA practices to study this and really looked at ancient humans all over Europe. We've looked at them in Africa, in Asia, in Australia, you name it, and really trying to really demonstrate that everyone has a unique history to their oral microbes. And a lot of that is based on the cultural practices and diet and environments of your ancestors. So if we know that information, then really what we might be able to do is translate that into improving health today. And so I was actually sitting one night chatting with my husband about my research and talking about all these cool ancient DNA projects we were working on. And he said, it's really awesome that you're reconstructing all of these ancient microbiomes, but when are you actually going to take that and use it to stop a disease, right? And stop somebody from having caries or stop somebody from having periodontal disease. And I kind of laughed it off at first, but then it really started eating at me and I started thinking about it. And it's like, how do we take this information and translate it into, improvements today in modern health? And when I say improvements, I really mean thinking about disease treatments differently, thinking about instead of removing microbes and killing them and getting rid of them like we do with toothbrushing and all these different, daily hygiene routine parameters, is really to actually introduce healthy microbes and try to replace maybe some of those microbes associated with disease with healthy ones. And so with that, the idea for oral microbiome transplantation was born. We shorthand it OMT. So you might hear us talk a lot about OMT, and that really comes from the field of fecal microbiome transplants, which is called FMT for shorthand. You may have heard about people doing fecal gut microbiome transplants to cure, for example, infections in the gut. And we're kind of leveraging some of that ideology, but doing it in a very different way, thinking about biofilm formation and dental plaque in the mouth, and really figuring out ways to take microbes from ultra healthy people who have never brushed their teeth, never been to the dentist, and never had any oral disease, and growing that up in a 3D printed flow cell that we invented, that Kevin is a whiz in the lab at running. And then taking that sort of output of that flow cell, whatever's grown in there, and it's quite disgusting, right? Growing a huge big layer of dental plaque in the lab, it's really gross, even from a healthy person, right? Taking that up, mixing it in hydrogels, and figuring out a way to use that as a mechanism to transplant into a recipient's mouth. And so far, we've done this in, for example, mice, and we think that we have evidence of caries prevention, at least I should say rats, not mice. Sorry, we've done it in rats. And anyway, in a model for caries in rats, we find that we do have the ability to prevent some caries. So we're really excited about the technology, and that's sort of a broad introduction to how the heck we got started into it and a little bit about what we've done so far.
Yeah, that's great. Thanks for sharing that, Laura. And guys, please leave your questions in the chat for us so we can answer them for you live. This is a really, really exciting project because what this could potentially mean is that we're no longer treating after disease has happened. We're treating patients before disease even has the chance to happen. Caries, could be periodontal disease. The potential implications and applications of this project is really, really profound. So I want you to have the opportunity to ask any questions that you might have. They could be technical questions. They could be scientific questions. You guys are talking to me all the time, asking me for some microbiology content on the channel. This is your opportunity to get your microbiology questions. We have a distinguished professor in the field here. So please ask anything that you have, and we'll be happy to address them in the chat as they come up. And so we have some people typing in chat. We have a thumbs up. We have some patient listeners here. We're appreciative of all of you joining us here. But yeah, let's let's keep things going a little bit. Kevin, do you want to talk maybe a little bit about your experimental setup and some of the things that you've been doing in the lab, and maybe that will help people come up with some questions to ask?
Yeah, sure. So what we do is we essentially we collect plaque from these ultra healthy people, as Laura said, and we put it into a 3D printed lab system. So it is essentially a artificial mouth model. So we've got media pumping through it, so a nutrient media to essentially help the microbes grow, and we essentially grow it up for about 10 days. And then we essentially just do 16S genome sequencing on the microbes to get an idea on what we've essentially grown. So that's our entire system as well. And a lot has been gone into actually developing the actual 3D system, because there's nobody else doing something similar to that we're doing. So
Yeah. How has it been working with those rats?
It has actually been really, really hectic. I think feeding them 5 times a day, starting at 6:30 in the morning, and then finishing at 11:30 p.m. I believe. But we had a team of 3 people, so me and another student and another research sort of assistant, going through the feeding cycles. But we were just camping out on campus essentially at some point, because I was the emergency contact. If something went wrong, I was called on to the premises, and I lived the closest to the actual lab itself, so it just made things easy. It was just easier. But yeah, it was interesting. It was interesting because we were feeding these rats a diet that was almost 50% sucrose. So yeah, they were pretty happy to see me.
Yeah, that's right. A classic college diet.
Yeah, pretty much.
Cool. Well, that's great. And Laura, how, I think we talked about this in the podcast too, but we can elaborate a little bit more. How, because this is an important component of the project, how were you able to find the right patients? Right, the right patients to find this microbial balance, because as we talked about, it's not just one microbe. You have to think about the entire community of microbes that make up a potential successful transplant.
Yeah, you're absolutely right that it's thinking about the entire community, right? We're not just growing one microbe from people and moving that across like you would be with probiotics. We're really reconstructing an entire microbial community. So in some of these folks, we have somewhere between 120 to 260 different species, right, that we're finding in their mouths and that we're able to regrow in the lab together. It's important you keep them together and grow them together because they enjoy being together. But yeah, it is difficult to find what we call ultra healthy donors, right? And so there was a really huge, massive effort with our team at the University of Adelaide. And that includes people like Sonia Nath, who's a recent doctorate at the University of Adelaide. And she works with 2 of our other PIs on the project, Lisa Jameson and Peter Ziln, who are also both at the University of Adelaide. And there was just a tremendous effort on Sonia's part to try to reach out to folks, both that were kind of connected through the dental school, but also were just part of the local community, to really identify folks who had never been to the dentist. And, people heard we were working on this project about like, oh, hey, somebody's collecting plaque. Like, you've never had a cavity, you should go get your plaque collected. And it kind of just, it spiraled from there for the most part. But what we end up finding in our really healthy donors is there's kind of 2 camps of people. One are the camp that literally they never brush their teeth. They haven't really ever thought about it. They haven't gone to the dentist. They just haven't had any oral health issues, right? And they just have this kind of miraculous oral health. And then the other camp are people who have great oral health, but it's because they brush 4 times a day, they floss twice a day, they use, like all of the implements that you could imagine, right? And we may have been sampling near a dental school, so I think that might tell you something about about dental school students. But there were kind of these 2 camps of people that we found had really good oral health, and in the end, we were really after the people who didn't brush, right? Who didn't ever do anything, because that might be a better donor, for people that for example, aren't used to taking care of their teeth or who've gotten oral disease by not having good hygiene regimens, right? So for us, we kind of focused on those folks. And a lot of people thought that they would also be the people that just had really healthy diets, right? Maybe these are the folks that never eat sugar, right? And they're like, on some sort of crazy healthy diet. But in fact, one of our top donors that we actually used in our animal model, the person's consuming almost 300 grams of sugar a day, right? So they're not on these super clean diets, right? They're, kind of regular old folks, right? That just have these amazing microbes. And that's really what we're trying to leverage here.
Mm-hmm. Great. And that's one thing that we've talked about in the podcast and on my channel as well, about this idea of caries being a multi-factorial process. It's not just having one thing. You have to have a host of things that line up together to cause a cavity to form. You have to have the right mix of bacteria. You have to have the fuel source, the food for that bacteria to secrete the acid that eventually produces the cavity. And of course, you have to have a vulnerable tooth surface. And I guess time too, because cavities don't form overnight. They typically take several years from an incipient lesion to one that you can actually see on a radiograph. And so all of these components together have to be true in order for a cavity to form. And so taking one part out of the equation, if we can take out the cariogenic bacterial community out of the equation, that's the hope. And that's what you're seeing in the rat model already, is that cavities are potentially not forming with the right mix of bacteria, which is super cool. I see we're getting some questions, which is exciting. I'm going to pop these up on screen. So if you guys continue to ask questions, we'll continue to answer them, because this is a pretty intimate live. We only have a couple of 26 people watching right now. So if you ask a question, good chances you're going to get it answered. So let's pull up this one. Nanoska asks, what about HSV? So we're talking about bacteria. We talked about caries, potentially periodontal disease, maybe later down the line in this research. But is there a potential relation to a virus? Herpes simplex virus is something we've talked about on this channel before. Do you think or postulate that modifying bacterial communities may have an effect on a virus, or probably not?
Yeah, it's interesting. I was thinking about this question completely differently. And so I can tackle it both ways.
Yeah.
So the way we've been thinking about this is really trying to avoid taking HSV from somebody who might be a really healthy donor in some ways, and actually, technically infecting a recipient with a human associated virus like HSV. You could also be thinking about, HIV or, all sorts of things, right? Take your pick.
Yeah.
And it's one of the reasons we really moved to this lab growth model and using the flow cell, because we grow it in the absence of any human tissue for 10 days. Really anything that requires human tissue to replicate, we're hoping is sort of weeded out of the system. And it really then just becomes these microbes that can grow, in this media that are dependent upon one another. The only thing that's really in that flow cell are these hydroxyapatite discs, which the microbes can sort of adhere to, once there's a pellicle formed. So for us, it's really more about avoiding these sort of infectious diseases that we might be, potentially putting people at risk for. And obviously, there's a lot of work that goes into making sure our samples are sort of free of infectious pathogens before we think about putting them, into humans during a clinical trial. But we think that 3D flow cell is going to be a really critical step, and not just sort of selecting for the right microbes, but also eliminating microbes that we would not like to have transplanted in somebody. But, to answer the question in the way that you were thinking about it, Dr. Ryan, I think we've just thought about caries and periodontal disease, but the sky might be the limit here. We know that microbiomes are, especially in the mouth, for example, are linked to things like oral cancers and halitosis and most things that have been examined at this date.
Yeah.
And so if we can manipulate the microbiome in some way, who knows what we're able to do, right? And I think for us, that's the really exciting part of this research is, we've picked these two incredibly common chronic oral diseases to kind of tackle first. but there's probably a lot of use cases for this. We're also really interested in how this OMT might actually impact the gut microbiome, right? And influence many other diseases that are linked to the gut. Things like obesity or type 2 diabetes, right? There's all sorts of things. So it's obviously ongoing research, but we're really excited about all the potential for many other chronic diseases as well as infectious diseases.
Yeah, that's great. And I see a lot of people joining the live. Welcome to everybody who is joining us. We're excited that you're here. And we're also getting a lot of really good questions. This is a live Q&A session, so please ask any questions that you have about this project as they come up. And we're going to do our best to address all of them, but we have some really, really good ones. So I'm going to start going through some more. Thank you, Nanaska. That was a really good, thoughtful question. This one's from Sahar. Is there any chance to work in the research?
Yeah, that's a great question. We get a lot of questions too about people who want to potentially even donate their plaque. These folks that they're like, wait, I'm one of those ultra healthy people, and I thought I was weird, but maybe I'm awesome, right?
I love that.
So we do have a lot of people reaching out. If you're interested in this type of research, please shoot me an email. My Penn State email is lsw132@psu.edu, and maybe somebody can throw that in the chat. We would love to sort of talk with folks about research potential, all sorts of those things. This is a very emerging area, and it's an area that we're actively seeking a bunch of funding into. So we're going to need folks who are interested in this to carry the torch and keep working on it.
Awesome. And we'll get that email and chat for everybody. And then how about we could talk about even maybe an easier way to get involved pretty quickly through conducting an interview with one of you guys, and how would that go about?
Yeah, absolutely. So, if you wanted to work in the research, that could be a little different. But if you are interested in helping with the research right now, you can totally do that as well. So one of the things that we're trying to do is figure out the best way to develop this technology so that it's useful in the clinic. And it's not frustrating, and it's not complicated, and it's not something that a dentist or a hygienist or a periodontist or even an orthodontist, right, doesn't want to touch because it's too frustrating and wasn't designed well. So we would love to talk to anybody in the dental profession. It doesn't matter where you are on the planet or what area of study, in the sort of dental profession you're in, right? Even if you're front of house, like we would still love to talk to you about, even the complications of like shipping this sort of product to your office, right? But we would love to talk to anybody, dental students, hygienists who are cleaning people's mouths and potentially could be administering this in the future, dentists who might have particular use cases, or they really want to have a hand in figuring out how to develop this technology, in ways that they might want to use it. We would love all of that feedback. And so Kevin and I, over the next 6 weeks, are going to be conducting 100 interviews, 100 different interviews of anybody in the sort of dental profession about this technology to get your thoughts and ideas about how we should develop it. So if you would be willing for us to ask you questions, we would love to do that. We would super, super love to. And so I know in the podcast, there's a link to, it's a bit.ly/bookomt link where you can schedule an interview with us. You can throw any time in there. Or if none of those times work because you're living in a different country, shoot me an email. Again, my email is there in the chat, lsw132@psu.edu. We'll figure out a time to meet. I don't care if I have to be up at midnight. That's totally fine, right? We'll find a time to meet, to have your sort of voice incorporated in this research. So we would absolutely love to talk with any of you about how you think we should design it. So yeah, if you're interested, we can also pop that link. I see. Yeah, great. The link now to sort of booking an interview slot with us is available. Whatever time works for you, we'll make work. We would love to talk with each of you and get you involved in the research straight away right now.
Awesome. Yeah, this is the link if you missed it. You just have to click on that. It's right in the live chat now, and then you can sign up there are a lot of open time slots. I give you both a lot of credit for having all of this set up. It's really, really well organized, and all you have to do is click one time slot and you'll get to talk and contribute. Because I see a lot of you are saying you're interested, you want to be a part of this project. That's a great first step in doing that. So make sure you click on that link and sign up while there are a bunch of slots available. And Laura, correct me if I'm wrong. That's your correct email address?
It is. Yep. That is my correct email address. It goes right to my Penn State email. and I'll get back to everybody about them. I promise.
Awesome. So you can reach out to Laura too, if you want to send her an email. That would be great as well. Good. Awesome. Well, let's get back to some of our questions. We had Sahar. That was a great question. Appreciate you asking that. We have a question from Kitty Kong. Strep mutans for enamel caries. All right, that's a great question. I'm gonna let Laura. I know Laura wants to answer this one, and I will say one quick thing. Yes, but there's a caveat. But yes, this is one strain of bacteria we talk about a lot on this channel as being like the main player in forming caries. But there is an asterisk there because it's not the only one. All right, Laura, I'll let you take it away.
Yeah, absolutely. Dr. Ryan's completely correct. And so strep mutans is the one, or streptococcus mutans, right? More specifically, is the species I think that people think of when they think about a microbe causing caries. And we know that this microbe, for example, can eat lots of sugar and produce lots of acid, right? And that essentially contributes to these caries in your enamel. The problem with it is that it hasn't always been that way. So in fact, we've done a lot of evolutionary analysis of this strain that we're getting ready to publish. And it turns out, even as recent as a couple hundred years ago, this microbe didn't have the ability to produce a bunch of acid. It kind of produced a small amount of acid. And it actually had gene duplications in its genome that allow it to produce more acid and also produce antibiotics that fight off other streptococcus species so that it can be the only streptococcus in the mouth producing lots of acid. We've also looked at this across many different indigenous communities around the world today, people who don't have what we would consider the industrial European descent microbiome, which is typically what's written about in the dental school textbooks. And folks living in different parts of the world with very different evolutionary histories, very different genetics, very different lifestyles, have very different oral microbiomes that are linked to caries. In fact, in ancient South Americans, we don't see any streptococcus species, right? And so we know that there's all sorts of unique microbes in the past causing caries there, because people still have caries, they just don't have streptococcus species. We know, for example, too, some work that we've done in Australia with Aboriginal Australians and Torres Strait Islanders, that there's whole cocktails of microbes, including lactobacillus species, Veillonelliaceae species, that also produce lots of acid, right? And in fact, might be more of a concern in some of those populations rather than strep mutans. We also know from Native American populations here in the United States that other streptococcus species, things like Streptococcus sabrinus, right, can also produce a lot of acid and be very directly associated with caries. So when we say strep mutans for caries, sure, in some people, but it still wouldn't even be the only player there. You still have to have that environment that also allows for that strep mutant species to grow to high levels and do its thing, produce lots of acid, right? And so what's happening in the overall microbial community that allows that species to happen, that's really what we're targeting, is if we can replace a bunch of these other coexisting microbes that sort of limit strep mutants from being able to do nasty things, then that's how we prevent caries. But that's going to be different in every population we sort of look at. This sort of industrial, usually of European descent microbiome is awesome. It's one cool microbiome, but it's one of many. And we're going to have to understand this in each population as we go. And one of the cool things about OMT technology is that you can get donors from any population, right? And you can use that donation within a single population. So, for example, we can get Aboriginal Australian donors and use that as what we as our basis for OMT in Aboriginal Australians, right? We don't have to take European microbes and put them in Aboriginal Australians, for example. So, that's keeping this sort of cultural signal in the context of caries development is really important to us. So yeah, great question, Kitty Kong. It's awesome.
Awesome. Great. Here's another good question coming up from Bertha HH. What good microbes are being transplanted?
Yeah, this is a great question. So the question is the answer is that completely depends. And so all of our donors are slightly different. None of them are the same. And so I wish I could say, yeah, if you've got these 4 microbes, boom, you're healthy, done, right? That's just not how it works. Some of our healthy donors have really high levels of micrococcus, for example. Some of our donors have really high levels of other taxa that we would have never expected to necessarily see in the mouth, right? And so there's other ways of building these biofilms across microbial species that look to be protective against disease. And so a lot of these, again, taxa, the things that you study in your dental school textbooks are still going to be there, but they're in the presence of many of these other species that we don't typically see. So there isn't like a beautiful cocktail recipe formula that we could just say, awesome, take these 5, transplant them. No, it's more like, we've got 5 different microbiomes across 5 different donors, and let's try to figure out which donor we should pair to which recipient to have the best effect.
Mm-hmm. Yeah, makes sense. It's a very careful process. It's not just one solution for everybody. Everybody gets this caries preventative cocktail, but it's tailored like an FMT, tailored to that individual patient. Great. I see a couple of questions that I'm going to pair together for the next one. Ninoska asks about pH changes, and so does Scarella, who asks, how do factors like pH affect potentially this process?
Yeah, you're completely right that pH affects this process. And it was something we were really interested in studying from the beginning. And so one of the things we do with all of our donors, and I'll let Kevin tell you all about the whole process of this in a minute, is test the pH of their plaque. We also look at the pH too after we've grown it in a flow cell. And we can do a glucose challenge of these microbiomes and understand how much these microbial communities are able to buffer the sort of signatures in the mouth. And we really think that, for example, good donors against caries are probably people who can buffer lots of acid in the mouth, right? They're people that sort of keep the pH high regardless of how much sugar you throw at them. And so pH is absolutely critical. But it might be that we need to sort of select for different types of communities that do different things in the context of pH when we're trying to solve other diseases, right? Just because, a high pH or a well-buffering microbiome and a glucose challenge might be good for caries, it might not be great for periodontal disease, for example. But pH is a really cool, cheap and dirty indicator, we think, of a phenotype or a physical process that we're looking at in these microbial communities that might be really indicative of whether or not somebody's a good donor or not for a particular disease status. Mm-hmm. Kevin, do you want to tell everybody about how you test the pH of a crazy biofilm in the lab?
Yeah, so it's essentially what we do is we take a little bit of media out from the actual model the 3D printer model that we have. So we do it across all 10 days, so literally starting from day 1 to day 10. And we test the pH using a pH meter. And that's done literally every day, like I said, from day 1 to day 10. And what we found is the pH usually hovers around 7, which is always a good sign. Because if the model is selecting for species that are producing high amounts of acid, obviously that's a bad sign, and that would be indicated with a very low pH reading. So far, we've not had any of those systems give low pH readings, so which is a good sign.
Mm-hmm. Great. And I'm just going to ask this question because I know some people in chat might be thinking about it, because we talk a lot about the critical pH of enamel, which kind of depends on the presence or absence of fluoride. And so maybe we could talk about that a little bit. Maybe what your considerations are in terms of that, the presence or absence of fluoride.
Yeah, that's a great question. We have been recording in what we call our metadata, which is just our information about our donors, whether or not they use fluoride on a regular basis, as well as whether or not, for example, they grew up in an area that had access to, for example, water that had that was fluorinated in it. So, I kind of being able to try to reconstruct somebody's history in the context of fluoride, but also whether or not they're currently using fluoride, I think plays a big role in sort of our ability to really understand how much those microbes are contributing or not to caries development, right? In the context of the enamel. And so it's something we've thought about a lot, but we're also thinking about it in the context of how you sustain an OMT after you might receive one. And so, if you're somebody who received an OMT, you've got your transplant, everything's going well, you go back to the, hygienist or dentist 6 months later for a cleaning, and they offer you a fluoride treatment, does that mean that you just sort of tried to wipe out all of those microbes that you just spent all that time donating into your mouth? And so, whether or not these things are exclusive, fluoride and OMT can't be used together, or whether or not they can be offset, or maybe one's used every other year, whatever that looks like, is something that we're thinking about long and hard, and something we're going to have to, I think, look at it in a clinical context. Because we are worried that fluoride treatment could just undo all of the OMT work that we just spent all that time doing.
Sure. Yeah, makes sense, because that's one of those things that's been hotly debated for a long time, is the use of fluoride. How much should be in drinking water, how much shouldn't be, and it'd be interesting to see that, in this context, does OMT take a similar role in the community, and how does that play in the future of dentistry? So really interesting to think about. Let's bring up another question here. We have a ton that are coming in. Thank you, everybody. Giselle asks, how does Crohn's disease affect the gingival tissue and oral health?
Yeah. This is a great question. And I would say it's still an active area of research, especially in the context of the microbiome, right? So we know shifts in the gut microbiome are absolutely associated with Crohn's. There's a couple clinical studies going on looking at fecal microbiome transplants for the treatment of Crohn's. And it certainly looks like matching a donor plus the right type of diet means likely, more FMT success in people who have Crohn's. And it's true in people who have Crohn's as well, that sometimes they will experience, for example, sores in their mouth, or inflammation, right? And I'm sure Ryan can tell you more specifics on that. And so, this could be, again, one of these other things that we think OMT might be useful for, right? If these oral microbiome transplants can do things that are, for example, anti-inflammatory, in the same way sometimes we think about some probiotics, some, definitely not all, some, if we can have sort of anti-inflammatory effects from that, it could be that it could help, treat some of those symptoms or something along those lines. Definitely still an active area of study and something that needs to be researched. But basically, because your digestive system is one big long tube, right? The same sort of things that are happening in the gut can be happening in the soft tissue of the mouth. And we separate those sometimes, right? And one's dentistry and one's medicine, and they're separate somehow, but they're not, right? And so thinking about how these things are linked and how they're connected and how the microbiomes are connected and linked too is really, really important.
Mm-hmm. Yeah, that's great. And I love that she brought that into the discussion about the digestive tract as being one long system, and it has effects on the entire body. The oral cavity, as we've seen, has a close connection and association with systemic diseases like diabetes, and we could, there's so many we could list. And it's funny because Crohn's disease was one of the, I release weekly practice questions on my Patreon page, and I actually had one not too long ago about Crohn's disease, because there was an orthodontic patient who presented to the clinic with this really demarcated linear ulceration at the base of their vestibule on both the right and left side. And I thought, oh, that's really interesting. Is that because of just some mechanical abrasion with the brackets or the wires? But the presentation was kind of odd, because usually if you have some bracket rubbing, it'll just be on one spot, or it'll be kind of directly outside of where that bracket is or where that wire is, not down at the base of the vestibule. So it's like, that's kind of a strange presentation to be that location, and then also to be symmetric on both the right and the left side. So anyway, thought more about it, and apparently these linear ulcerations are a sign of Crohn's disease. There's a very specific presentation of it in the mouth. And so because it's anything, once you get to something autoimmune, it typically has some like very strange, unique presentation. It turns out that's one of those. You also mentioned a lot of ulcerations. That's something that we see a lot too. So gums are more susceptible, not necessarily to like periodontal disease, but certainly to other soft tissue conditions like ulcerations, and things of that nature. So yeah, great question, Giselle. Cool. Let's see. Trying to go back to where we were. Okay, we answered the pH. Okay, here's one from Ice Age Revenge who asks, any idea yet on how you would, I love the name by the way, on how you would ultimately select for certain bacteria without harming all good bacteria?
Awesome name. I love Ice Age Revenge. It's great, especially for all the ancient DNA work I do.
Yeah, there
That's
you go. Yeah,
super
you
awesome.
really would appreciate that that name.
Yeah. Yeah. So yeah, how you select for certain bacteria without harming all the good bacteria. I guess the short answer is in our technology, what we're doing is we're placing most of the microbes in the mouth, but not all, right? And so we're putting this donor set of microbes into a recipient. And at least in our in vitro models, when we did this, we saw that the recipient's microbiome after transplantation kind of is a new microbiome. It's not really just the donors, it's not the recipients, it's this new microbiome. And what it really is this sort of mixture between, it's mostly donor, but there's some recipient microbes that still stay there in the process. Now, we spend a lot of time trying to remove the recipient's microbiome, right? Because this microbiome is hopefully associated disease and something that we want to get rid of. It's not something that we want to keep around, right?
Yeah.
We spend a lot of time thinking about how to get rid of that. And our best sort of strategy right now is sort of we would do a planing and a scaling and a full cleaning and polishing. And then we do a couple different chlorhexidine type treatments to really remove any of the existing recipients' microbes before they then get an OMT. But despite all of that, some of those microbes survive, right? And they're still there. It's not like they're completely gone or completely independent. And so, really, at the end of the day, after an OMT, the recipient's microbiome is really kind of a new, and it's specific to them, right? Because it has this sort of recipient signature in it. It really is this new type of microbiome. So we don't really have the ability right now to very specifically select for, just keeping the good bacteria in a recipient and getting rid of only the bad. What we're doing is really trying to get rid of everything and hope that some of the good ones stay with this new donor set of microbes, which is hopefully supporting a healthy environment in the mouth such that it would also support those good species staying around. But there's a lot of, again, active research going on in this space, and really understanding what that, new recipient microbiome looks like and what its sort of long-term consequences are, is something we're really interested in.
Awesome. Great. Well, I'm going to keep it moving because we have about 20 minutes left and we have a whole bunch of questions. We really appreciate all of your engagement with the panel here. So thank you again for all of you submitting questions and listening intently. We really appreciate it. This next question comes, I just want to make sure I don't miss any. Good. Okay. Next one is coming from Karthik, who asks, what do you think would happen if an oral cavity is completely devoid of microbiomes? Did you zero in on any one beneficial microbe which could overtake the other harmful ones? Interesting idea, thinking of completely being devoid of all bacteria. What do you think about that, Laura?
Yeah, gosh, that's a really great question. That's really got me thinking. That's an awesome question. There are, we call them germ-free mice. So there are mice that have been bred essentially in a bubble, in an area without any other microbes. And so the grandparents or, the previous generations of those mice have gone through a lot of antimicrobial treatment to try to wipe out their microbiomes. And these germ-free mice that sort of live in the bubble, you can then do all sorts of really interesting research things with them, like, for example, humanize them in the context of the microbiome. You can insert human-associated microbes in them. But before you do any of that, these germ-free mice really have very few microbes. Now, they don't have none, which is what I think people were hoping that they would have. They do kind of have a couple of their own. But they're probably the closest thing we have to having mice that are devoid, of an oral microbiome. And so I don't know of a lot of research that's been done on germ-free mice in the oral microbiome, but that would be a super cool area of research and something that, might be fun for us to try out an OMT in, just to see what the heck happens in the context of, does it put the mouse at risk of infection or risk of other types of diseases when you do introduce this huge big bolus of microbes in a space that otherwise doesn't have any, right? So I think that it's a really great idea, Karthik. Awesome sort of research idea there. But yeah, I'm not sure like what I think would happen, right? I think the oral microbes that we'd put in would just set up shop, right? And probably take place. The hope is though that they have enough of an immune system developed to be able to, have that balance with the microbes that you're putting in. So a lot of these germ-free mice, they have very bizarre immune systems because they didn't have the microbes to train and educate the immune system when the mouse was developing. So it's also kind of it's an interesting part. If you didn't have any of these microbes, what would your immune system look like, right? Would it be able to respond in ways that we need it to respond? So yeah, I don't know if that answers fully the question, but that's where my thoughts are anyway. And I think it's a great research question.
Yeah, no, it's fascinating to think about. Going back to this equation I typed in the chat, it's very, very simple, oversimplified, but the combination of these elements causing caries. Like for instance, let's just focus on the tooth for a second. One of the benefits to placing a dental implant is that you don't have to ever worry about it getting a cavity, right? Because there's no enamel, dentin, or pulp. Now, implants are not immune to other oral conditions like gum disease. We don't call it periodontitis, but we call it peri-implantitis, but similar, but slightly different. And actually, interestingly, Laura, you may know this, that there are different bacterial communities that tend to facilitate that disease process in contrast to periodontal disease. So super interesting. But with a dentulous patient who's in dentures, of course, they have their own host of things to deal with there, but cavities is not one of them. So you remove teeth from the equation, can't get cavities. If you remove, this is my theory, if you remove all food from bacteria, and correct me if I'm wrong, Laura, I could be wrong, and I don't think this is really possible. Oh, we have the best. Wait. Okay, hold on. I just want to stop. That was great. Kevin, you can introduce our other secret panel member.
Yeah, literally just.
That's fantastic. That's great.
That's Murphy.
That's amazing. I love that. So, but anyway, my theory is that if you take away all food from these bacteria, they cannot produce cavities. Maybe, I don't know. I don't think it's realistic to say you cut out all carbohydrates or all simple sugars or whatever feeds those bacteria. But would you say that's true or not? I'm just postulating. I don't know if that's actually true or not.
Yeah, absolutely. I mean, it's kind of a weird experiment to do. Oh, hi, Murphy. It's kind of it's a very interesting experiment to do, right? And I think that you're right in your postulation. Like that would kind of be along the lines of how we would think about it. Microbes have to have many of these diverse nutrients. They have to have lots of micronutrients as well to grow. And if they don't have them, they simply, oh, Murphy's so cute. If they don't have them, right, they simply can't grow. And in order for you to get diseases like caries, these microbes essentially need to grow to high levels and grow kind of, we call it out of control is what we're saying, shorthand in the lab, right? That those microbes have grown out of control, right? And so without the nutrients to grow and to replicate and to do that, yeah, they're not really, I think, going to be able to cause disease in many of the same ways that we would have normally, thought about this. That's a great idea.
Cool.
Yeah,
awesome.
Oh.
The star, Murphy, the star of the show.
He's interested in OMTs.
That's amazing. All right, great. Next, we have Sahar returns with another question here, who asks, can changing the oral microbiome help in decrease caries in susceptible individuals?
Yeah, I've been talking a lot. Kevin, do you want to take this one?
Yeah. So yeah, that is essentially the main idea behind the OMD. It's essentially to decrease to target people that are more susceptible to dental caries. So yeah, definitely it can definitely have an effect, a positive effect. That is. So
yeah, one of the things we're we're really thinking hard about is which population of folks we do our first clinical trial in. And certainly folks who experience caries, very quickly and very drastically are definitely kind of on our list of people to test. But there are also situations, right, where people's risk for caries development drastically changes. For example, if somebody has radiation therapy and is experiencing mucositis, right, that might be a time when somebody's caries risk has drastically shifted, right, and they might need more tools in their toolkit to try to prevent caries. And so, we're thinking a lot about what are the best populations to test this in, people that can potentially receive the most benefit from this technology the quickest. We're also thinking a lot too about, folks who may have difficulty in maintaining their hygiene of their health. For example, in, aged care facilities or, different areas like that, where OMT might be kind of a saving grace because they might not have to, be as concentrated in their hygiene regimen as they were before. So, these are sort of ideas that we have and we're thinking about. I also think a lot about kids and whether or not OMT could be useful, for example, in children experiencing, for example, severe early childhood caries. Obviously, this research would need a lot more work before I feel comfortable, putting it in children for the first time, because changing the development of their microbiome might have, other consequences later in life. But for kids who are experiencing incredibly severe caries, maybe that's something that you look at, right? And that the risk outweighs the benefit, for example. So there's all these populations that experience very severe caries, and that's really where we're thinking about OMT. Potentially having a best benefit is these people who are very susceptible to caries for one reason or another.
Mm-hmm. Yeah. So, and that's a great answer to Sahar's question. I want to highlight one word that I like that you use, Sahar, and that's susceptible. And as an orthodontist, my mind immediately goes to all of my braces patients who are automatically, just by placing appliances on their teeth, more susceptible to caries. Not because they automatically forget how to brush their teeth, although sometimes, honestly, that's how it feels to me. But really, in reality, it's just creating a magnet to which food and other things get caught and stuck to. And those people are at a much elevated risk of getting cavities because of all these nooks and crannies that become more difficult to keep clean. And so I could imagine in a patient with braces on, this could be potentially very utilitarian, because now instead of saying we're 2 years or we're 1.5 years into treatment, we almost got the bite fully corrected, but there are all these white spots around your brackets. They're about to become cavities, and we have to take these brackets off because I'd rather you have healthy teeth and slightly not fully corrected bite than you have a perfect bite and now all your teeth have cavities and you have to get them all filled. So if that could potentially prevent that, and we could keep the brackets on a little bit longer to get everything done, that would be pretty cool. So great question. Great question. Kitty Kong, thank you. Learned something new today. Well, that's great. We have a lot of learning going on here. We appreciate that. That's really great. Good. Let's see. I'm looking through if there are any other questions. Looks like we got most of the questions addressed. We have about 5 minutes left. So if you have any last questions for Kevin or Laura, please leave them in the chat here.
I think there's a question. Do you do in-study phase 1 in animals?
Oh, yes. Thank you, Kevin. Then Oscar comes back. Do you study? Yeah.
Yeah, so this is something that we've been discussing more recently. Obviously things like periodontal are huge problems in our pets, right? Mr. Murphy maybe might have some of that, who knows? But especially in our domesticated pets, it's interesting to study the evolutionary history of that as well, because these animals that are domesticated eating lots of human industrialized food are the ones that are more likely again to get oral disease. So we're interested in the connections there, but at the end of the day, this technology could be really useful in our pets as well, right? And it's interesting to me because there actually has been a study published where they've looked at OMT in dogs suffering from periodontitis, and they did it very differently from how we do our OMTs. And so there they were kind of taking saliva from one and kind of, using that as the sort of transfer material, whereas we're focused on the dental plaque and the biofilm and that really big, large substrate, that amount that we can sort of transplant. But in that study, they showed that and they only had one donor, and the donor wasn't really, selected or tested or examined. It was just like, hey, this is a healthy dog. So, from that perspective, we think that there's a lot to be improved upon. But in that particular study, they saw that it changed the recipient dog's microbiomes for about a week, and then it kind of went back to their original states. But I just think there's so much that we can do to iterate on that. But the study was just really great in our mind because it opened the door for us thinking about what this looks like in pets and animals, and really thinking about how we, take research and we iterate on each other's ideas and we move it forward, right? And so, that work was done by some folks that are now at the University of Adelaide as well, but also folks in Germany and all over the world. And so it's been really interesting to see how other people would approach OMT. And, as published results are coming out, for pets or things like that, what folks might find. But I think the bigger issue of doing an OMT in an animal is really, how we're going to deliver it. I think in a human, we can put it in these, mouth guards or sort of trays and then let it sit in the mouth, for 30 minutes, really let those microbes adhere. Whereas, in a pet, you can paint it. You could kind of paint a hydrogel. This is how we do the rat experiment, right? Is we paint the hydrogel on an anesthetized rat's teeth. And we do that once a day for 3 days. So you might be able to do that, but that might be, difficult, for different reasons, to say the least. And so we have to think about, how we get it to a pet, but maybe putting it in some sort of tasty hydrogel that they want to, keep licking or putting in their mouth or something might be a way to do it. Who knows?
Yeah, absolutely. I want to highlight, we're getting close to the ending of our session here. I want to highlight a great question that my brother Derek posed. He's helping to run the chat in this live stream. And he's wondering about how the microbe community, the oral microbiome, might be changed for someone who's using a feeding tube, where food is bypassing the oral cavity completely. What do you think? What kind of effect do you think that might have on the oral microbiome, Laura and Kevin?
Yeah, that's a really great question. I mean, I'm not completely up on the sort of oral microbiome research of folks with feeding tubes. So if there's new papers out there, apologies. But my supposition would be that their microbiome is completely different. And we know some of that probably through folks, for example, who are mouth breathers at night, or people who have, for example, like CPAP machines. We know that can have a slight impact on the types of microbes that you have in your mouth. So I'd have to imagine feeding tube also is the same way. I also think about kids and people who are also intubated, for example, for different reasons, one way or the other, or people who, we've been thinking about this in the context of very young infants, right, who have to undergo surgery too, and kind of similar processes happen. And, having ways to really support good microbes and avoid infection in those sort of situations too is really, really critical. So I think OMTs might totally be relevant there, but for different reasons, right? It might be to, say, block a secondary infection rather than keep the person from getting caries or something along those lines.
Right. That makes perfect sense. Cool. Let's see. Good job. Very enlightening session. That's awesome. We're glad you enjoyed it. Adamola, great to have you here. Looks like we have one last question that I'll address here. Roberto asked, what are the most common microorganisms in an endodontic infection? Thank you for the answer. I'm comfortable taking this one, Laura, if you want me to.
Go for it, Dr. Ryan.
So this, and again, I might be oversimplifying things because, again, as with everything that we're talking about here, this is in reality a microbial community that's responsible for an endodontic infection. It's not just one microbe, although I know a lot of the teaching I do is for these board exams where they ask you, like, what is the main etiologic agent of X? And so that's a lot of what I teach, is kind of the simplified version of things. So in that vein, I'm going to give you a very oversimplified answer to this question, Roberto. In primary endodontic infections, so that's a tooth that was previously unaffected, their pulp was previously healthy, has now become diseased, whether that's reversible pulpitis or irreversible pulpitis or symptomatic apical periodontitis. The primary etiologic agent in terms of microbe is going to be your Bacteroides species, the Bacteroides family. For a retreated endodontic tooth, for a failed endodontic treatment that needs retreatment, it tends not to be Bacteroides that's the main etiologic agent, but actually, kind of gross, the Enterococcus faecalis tends to be the main etiologic agent for that one. So that's what I would know for the board exam, if that's how you're asking. It's a great question. Laura, how did I do?
No, I think it's totally awesome. Great job. But one of the things I would just say is like, it's got an asterisk again, on those answers. And the reason I say that is because, for many years we relied most of our understanding or knowledge on this relied on being able to grow microbes from, biopsies from these infections or from, what was pulled out of the infection. Right. And we know today that we can only grow about 2% of the microbes we know of, right?
Yeah, right.
And so that really limits our understanding of how these things are formed, what's actually working together during an infection. So, there's lots of work going on to really use genetics and use these high-powered, high-throughput DNA sequencing approaches to describe whole communities that are happening during infection, rather than relying on culture or the assessment of just a handful of microbes.
Mm-hmm. Yeah, great. Fantastic. Well, we're closing in on the end of the session. I do want to get to this one. I think this is an interesting one. And Diana, please provide, if you have more details to the question, just so we know we're addressing it properly. But I think this is a really interesting question. Diana asking, is there advantage that some race has to contribute to the microbiome? Potentially, maybe some ethnic implications in terms of microbiomes and things like that. Is that something that we're even able to consider in this project? Is that outside the scope of this initial research? What are your thoughts on that?
Yeah, this is an amazing question, Diana. It might be my favorite question of the night. And I'll put my anthropologist hat on to really answer it. There's been a lot of people in the field that have suggested that people from indigenous communities or non-industrialized areas might hold the key to sort of solving diseases in industrialized countries, because their microbiomes are very different and they're not yet touched by industrialization. And so there's even been kind of calls to harvest and to catalog these microbes from indigenous people. And that immediately makes me incredibly worried, because we need to be doing research with indigenous people for their benefit, not just the benefit of people in industrialized countries. But that said, if researchers can work with communities and work with indigenous communities and really honor their wishes and their desires, I think that we have a whole heck of a lot to learn from the microbiomes and people who are not in industrialized countries. And industrialized is such a loaded term. There's a lot of things that we're assuming about that term that we absolutely shouldn't be, but for lack of having time to go into detail of that. But I think that, indigenous people have a lot to contribute to our understanding of how microbes are linked to disease. And I think that there's a lot to be done in this space, to really empower indigenous communities, right? If somebody has the key, has the specific microbiome that can cure, all caries, right? Then, more power to that community to have ownership over it, right? And to be able to work with developers and companies, right, to be able to commercialize that and benefit from it. The benefits really need to be, obtained to all people, and not just, the folks that live in countries of higher gross domestic product, right? So I think the answer is really yes, but it's a complicated one. But it's a really, really great question, Diana. Great question to end the night.
Yeah, thank you so much, Diana, and thank you to everyone who participated in this Q&A. I really enjoyed this. This was a ton of fun. I always love interacting live with our audience, and thank you for all the thoughtful questions. This was really great. So we'll end with just one more reminder that if you have more questions, or if you want to get more involved on a personal level and provide your own feedback and thoughts about this project, here's your time to do it. Click on the link that we have on the screen, and we're going to just repeat that in the chat. This is so you can sign up for an interview, so you can get that going. We only have a few more. Is it a few more weeks, Laura, that that's going to be available?
Yeah, we have essentially 5.5 weeks from right now. And so
Okay.
we would love to have your thoughts and your opinions about how we should develop this technology, who we should study it in first, anything that you want to share with us. We would absolutely love to hear from you. So please, please, please sign up. We would love to talk to you about it.
Awesome. Well, that will officially conclude our Q&A. Thank you to Laura. Thank you, Kevin. And thank you, Derek, behind the scenes. Until next time, guys, don't forget, genetics live class coming up on Saturday. If you haven't signed up, I have 3 seats left, just FYI. Do that because that's coming up very soon. But that's it for this week for Mental Dental Events. It was a busy week, but a lot of fun. Thank you, everybody, for participating. And until next time, take care. Bye-bye.
Bye.
See you, everybody.